Rebecca Henning, and I'm a life sciences educator at Abbott's College in Cape Town. And what we're going to do today is some life sciences revision for paper 2. So let's remind ourselves what topics are in paper 2. So we've got DNA, the code of life, meiosis, genetics and inheritance, and evolution by natural selection, as well as human evolution. As you can see from the table, the most marks are for genetics and inheritance and evolution. So those are the two topics that I'm going to focus in on today for our revision session. And we're going to make that really clear to us because we know that that's where we can score marks in the exam. So please do make sure that you study DNA and protein synthesis and meiosis as well. But let's do some genetics and evolution revision now. So firstly, let's talk about a monohybrid cross. So in genetics, a monohybrid cross is where we look at the inheritance of one characteristic. So just one is what we're going to look at. And we're going to do it through this particular example. So in dogs, rough hair, capital H, is dominant to smooth hair, small letter h. A heterozygous, rough-haired dog is mated with a smooth-haired dog. Represent a genetic cross to show the phenotypic ratio of the puppies. So we've been given some information here about a characteristic, and we are going to look at how this is inherited from the parent dogs to their babies, the puppies. And we are going to do this through a genetic diagram. So this genetic diagram has a particular format that you need to remember and know how to do, but a question like this would be worth 6 marks. So if you can remember the format and then you can use the information given to you in the question, you can certainly score all of those 6 marks. So in this case, we are looking at a monohybrid cross, so just one characteristic. And in this case, our characteristic has 2 alleles. So there are 2 variations of the gene. And so we've got the rough hair gene, which is the capital H. So, and we've got the allele for smooth hair, which is a small letter h. So in this case, our one allele is dominant over another. So the variation of the gene that allows for rough hair, the capital H, which is represented by a capital H, that is the dominant allele because they gave it to us because they told us in the sentence that it's dominant, as well as the fact that it is represented by a capital letter. So whenever an allele is represented by a capital letter, it is the dominant allele. So rough hair is dominant over smooth hair, small letter h. So smooth hair is the recessive allele. And this is an example of complete dominance. So this is the case where if a dog has one allele for the rough hair, the capital H, that is what's going to show up in their phenotype or their physical appearance. So if they've inherited just one copy of the rough hair gene, then that dog will appear having rough hair. So for example, like the dog in the left-hand side of the picture. Whereas in order to inherit smooth hair, a dog would need to inherit 2 copies of the recessive allele in order for that characteristic of smooth hair to show up in the phenotype, which is the physical appearance of the dog. So for example, the dog on the right-hand side has smooth hair. Right, then what have they told us about the parents? They told us a heterozygous rough-haired dog. Heterozygous meaning, hetero meaning 2 different alleles, is mated with a smooth-haired dog. And now we know that in order to inherit smooth hair, one has to have 2 copies of the recessive allele, the small letter h, which in this case is the homozygous parent, because homozygous, homo meaning the same. So that smooth-haired dog will have 2 copies of the same allele. So from this information, we've already managed to work out the phenotypes and genotypes of the 2 parents. Right, now let's put that in our format for our diagram. So we start off with the P1 generation. So P1 representing the parent dogs. In this case, we were given the phenotypes in the question, so we were told that there was a rough-haired dog that was mated together with a smooth-haired dog. Then we can work out the genotypes of the parents. So we were told that the rough-haired dog parent was heterozygous, meaning having 2 different alleles, and we know that rough hair is represented by the dominant allele, which is shown through the capital letter H, and then because it's heterozygous, it's 2 different. So the parent will have 1 dominant allele and 1 recessive allele. So this parent will be capital H, small letter h, which is then crossed together with a smooth-haired dog. Smooth hair is a recessive characteristic. We were told that in the question. And so in order for smooth hair to appear in the phenotype or physical appearance of that individual, they need to have inherited 2 copies of the same allele. So in this case, the parent would be homozygous, having 2 of the same alleles, which are 2 small letter h's. Right, so just from the information in the question, we can already figure out the genotype and the phenotype of the parents. Then what happens? Well, inside the parent's body, in their reproductive organs, what happens is meiosis takes place. So meiosis is cell division in order to make gametes, which are our sex cells, which is sperm in the case of a male and eggs or ova in the case of a female. So these 2 dogs have undergone meiosis, they've made gametes, and then those gametes are going to meet. So when these 2 dogs mate with one another, the sperm and the egg will fuse together and fertilization will occur. All right, so we've already got a couple of marks here. We got a mark for saying P1. We got a mark for giving the phenotypes of the parents, a mark for giving the genotypes of the parents, and we also get a mark for writing meiosis, gametes, and fertilization. So 4 marks already. Sure, we can do this, guys. All right, so now we need to work out what are the gametes that these parents are going to produce. So in the case of our rough-haired dog, they can produce sex cells with either a capital H in them, a dominant allele, or with a small letter h in them. Then our smooth-haired dog can produce gametes with a small h or the other small h. So those are our options. Now we draw something that's called a Punnett square. And a Punnett square is where we are going to put these alleles in, and we are going to look at if this sperm and this egg were to fuse together and fertilize one another, what alleles would that puppy get? What genes would that puppy inherit from its parents? So we're going to draw our Punnett square, and what we do is it is a block. It has 9 parts to the square, 9 blocks. The top left-hand side block we cross out because we're not going to write anything there. Then in the left-hand column, we are going to write the alleles for the one parent. So in this case, the gamete options for this parent are a capital H, dominant allele, or a small letter h. And then across the top row of this table, we are going to write the 2 gamete options for the other parent, which in this case are small letter h and small letter h. All right, now we put this information together. So what we're going to do is we're going to go from the capital H to the small letter h, and we're going to put this information together. So if that sperm and that particular egg meet and fertilize one another, the puppy is going to inherit a dominant gene, capital H, and a recessive gene, small letter h, from the parents. And we do this over and over in order to complete the table. And so what we can see here is then we're going to look at the F1 generation. Oh, I've just realized there's a mistake in my table, but don't worry, we're going to fix it. So if we look at the bottom right part of the table, you can see that there should actually be a small letter h and a small letter h in that block. I hope you guys managed to pick up on that and notice that. All right, so now we're going to go to the F1 generation. So this is our first filial generation, and these are the puppies. So these are the babies from these particular parents. And what can we see about the genotypes of these puppies? Well, we can see we're going to have two puppies with the genotype of capital H, small h, and we're going to have two puppies with the genotype of small h, small h. So we're nearly done. We got this now off our Punnett square, and now they asked us to talk about what is the phenotypic ratio of the puppies. So now that we know what the possible genotype options are for the puppies, we can write down what the phenotype options are. So in a monohybrid cross, where we're looking at inheritance of one characteristic, we are going to have 4 possible genotypes for the offspring, for the babies. And in this case, our 2 puppies that would inherit the capital letter, small letter, they are going to have rough hair because they've got that dominant allele, the capital H for rough hair. So that is what's going to show up in their phenotype. And then the 2 puppies that inherited the 2 small letter H's, those are going to be 2 smooth-haired puppies. And so we're going to end up with 50% of the puppies having rough hair and 50% of the puppies having smooth hair. All right, so we've now answered this question and we've scored ourselves 6 marks. So there are a lot more marks available for this question than you can actually achieve. So there's actually about 8 or 9 possible marks that you can get here, but they can only give you up to a maximum of 6. So please, very important to remember this formatting. Practice using this format for answering the question. It will really help you. And you can even get some marks just for using the correct formatting, even if you end up getting some of the alleles and genetic part of the question wrong. So please practice your monohybrid crosses. Very, very important. You do also need to know a little bit about dihybrid crosses, which is the inheritance of 2 characteristics. And you don't need to be able to draw the Punnett square for that, but you do need to be able to understand the inheritance of the 2 characteristics. Right, let's have a look at a pedigree diagram. All right, so a pedigree diagram is like a genetic family tree. So it's showing you the phenotypes of the individuals in this particular family. So in this case, if we look at our diagram here, it is showing us the inheritance of hemophilia. Now, hemophilia is a disorder that is to do with the fact that you don't produce the clotting factors in your blood. And so if you suffer from hemophilia, what happens is if you cut yourself, it's not going to stop bleeding. So it can be potentially life-threatening and dangerous. So it's important for us to understand how it's inherited and which members of the family suffer from hemophilia. So if we look at this pedigree diagram, we can see that we have 3 generations of this family shown. We've got the grandparents, which are numbers 1 and 2, and then they had children, which are 3 to 7, and then those children had their own children, which is the 3rd row from 8 to 14, number 8 to 14. So that is all of our parents, grandparents, parents, and children, 3 generations. The pedigree diagram, important to note also, always represents a male as a square. So if you see a square on a pedigree diagram, it is always a male. If the square is not colored in, so it's white inside, that means that that is a normal male, and that is a male that does not have hemophilia. If the square is colored in or black inside, then it is representing a male that suffers from hemophilia. Then, females are represented by circles. Again, a white circle or a not colored in circle is a normal female. And a colored in or black circle is a female that suffers from hemophilia. So we can tell all of that information just from looking at the picture. All right, now what else are we told in the question? Well, we're told that the pedigree diagram shows the inheritance of hemophilia in a family, and the allele causing hemophilia is represented by capital X with a small letter h in superscript above that. And the normal allele is represented by capital X, capital letter H in superscript above that. So what this is telling us is that hemophilia is a sex-linked disorder, meaning it is related to whether the individual is biologically a male or biologically a female. Reminder, I'm talking now about biological sex based on your genetics. I am not talking about gender, which is a social construct. Right, so they've actually told us quite a lot here by telling us that the hemophilia is a sex-linked disorder and it is inherited on the X chromosome. So we know genetically a biological female has 2 X chromosomes and a biological male will have 1 X chromosome and 1 Y chromosome. Right, our first question asks us to determine the phenotype of individual 4. So this we can actually get straight off the picture. So if we have a look, where is individual 4 located? So we can see in the middle line, middle row there, we've got individual 4, and it's a circle and it's not colored in. So that tells us that individual 4 is a normal female. There we go, we've answered a question already. Right, next, what is the genotype of individuals 1 and 2? All right, individual 1, we can see is a white square, making it a normal male. All right, so if it's a normal male will have 1 X chromosome and 1 Y chromosome. So in this case, the X chromosome will have the normal allele on it. So it will be a capital X with a capital H in superscript above that and a Y chromosome. So X, capital H, Y for the male. Individual 2. So individual 2 is a normal female. So a female without hemophilia. But if we have a look at individual 2's children, we can see that children number 6 and 7 have hemophilia. And in order for them to inherit hemophilia, there must have been hemophilia somewhere in the family. So what we can tell is actually that individual 2 must be heterozygous, meaning she will have 1 normal allele and 1 allele that causes hemophilia. So this allows her to pass that allele for hemophilia on to her children while not suffering hemophilia herself, because the dominant allele is going to mask or cover up for the other allele. So individual 2 will have the genotype of X capital H and X small letter h. And from this, we can then explain why females have a smaller chance of suffering from hemophilia. So this is what I was saying about how hemophilia is caused by a recessive allele, which is carried on the X chromosome. And so because females inherit 2 X chromosomes, if they have 1 X chromosome with the normal allele and 1 X chromosome with the recessive allele, the normal chromosome will mask or cover up for that. So the female can only be hemophiliac herself if she has 2 copies of the recessive allele. So in this way, this female is what we call a carrier. So she carries the gene, but she does not suffer from the disease herself. Whereas a male only has 1 X chromosome. So if they inherit that X chromosome with that small recessive allele on it, unfortunately, they're going to have hemophilia because the Y chromosome is totally different and cannot cover up for that X chromosome. All right, so that is a pedigree diagram. They love to ask interpreting a pedigree diagram type questions. And then we've also touched on looking at a sex-linked disorder where the disorder is carried on the X chromosome. So in this case, they told us that actually in the question by showing us the capital X with the superscript letter on it to represent the alleles. They also might just say it in words. They might actually explain that. And then when you're talking about the genotypes, you're going to have to talk about X and Y. All right, so keep that in mind. However, you can also use a pedigree diagram to represent genetic diseases and disorders that are not sex-linked, so not related to whether you're a male or female as well. But remember that you can always work out what's going on from the diagram because you know that males are squares and females are circles, and you can look at how characteristics have been inherited from parents to their children. All right, so we're going to take a step now towards evolution. So again, evolution, very important section, makes up a lot of marks in this paper. And it's so important that we understand how this process takes place. So we start off, first of all, with Lamarck's theory of evolution. So Jean-Baptiste de Lamarck came up with this initial theory of evolution. We've since realized that Lamarck is completely wrong, but it was a pretty good idea at the time. And he was the first person to really suggest an idea of how this took place. So Lamarck proposed 2 parts of the story. He had a law of use and disuse, and then the law of inheritance of acquired characteristics. So the most popular example is Lamarck's giraffe. They love using this example. So what happens is originally all giraffes had short necks. According to Lamarck, these giraffes with the short necks had to keep stretching their necks up and up and up, trying to reach the juicy leaves on the higher up branches on the tree. And because they used their necks so much by stretching them, that meant that the size of the neck increased, and the neck got longer and longer over time. Then these individuals were able to pass on that acquired characteristic of the longer neck to their offspring. So unfortunately, Lamarck is wrong because this is actually based on genes. And so a good example would be, let's say you go to the gym and you do lots of weightlifting at the gym and you get really buff and lots of big muscles. Now, when you reproduce and you have a baby, is your baby going to be born with these giant muscles? No, it's not. And that is because you've just changed your physical appearance. You haven't changed your genetics. You haven't changed your DNA inside. And the genetics are what is passed on to the offspring. So you cannot pass an acquired characteristic on to your baby. All right, so that was the initial theory of evolution. Next comes Darwin's theory of evolution. So Charles Darwin, famous English naturalist, proposed the idea of evolution by a process called natural selection. And so this is what our current theory of evolution is based off of. So we'll illustrate this by using an example. So a saguaro cactus is a plant that has the ability to grow in desert conditions because it can store water. Within a population of cacti, the stems can be smooth or pleated. The pleated stems have the ability to store water for longer periods of time. Explain why in a desert there would be greater proportion of saguaro cacti that have pleated stems than those with smooth stems in the population. So this question is actually asking us to explain evolution by natural selection, because what we're seeing is a change in the population over time. So evolution is this idea that organisms haven't remained the same. Organisms have shifted and changed over time. And this happens by this process of natural selection, where certain genetic characteristics give a particular individual an advantage to survive in a particular environment. So if we have a look at the picture on the screen, that would be a cactus that has the pleated stem. And so the pleated stem is able to store more water than those individuals that have the smooth stem. All right, so within a population of these cacti, some individuals have smooth stems and some individuals have pleated stems. So we see genetic variation within the population. So genetic variation within the population of cacti. Some have smooth stems, some have pleated stems. Those that have the smooth stems are unfortunately not able to store water for as much time. Therefore, they dried out and they died. Because those individuals died, they couldn't pass on their genes to the next generation. However, the cacti that had the pleated stems can store water for a long period of time, which means they have a much better chance of surviving in a desert environment. And because they survive, they are able to reproduce and pass on their genes for that pleated stem to the next generation. And so this happens over and over and over, with more and more individuals with pleated stems surviving and then passing on those genes to their offspring, to the next generation. And so over time, we're going to see more and more individuals in the population having a pleated stem as opposed to a smooth stem. So this is the idea of natural selection, where a genetic characteristic, in this case pleated stems, gives you an advantage, better allowing you to survive in a particular environment. So in this case, pleated stems work better in a drier environment because those individuals are able to store the water for a much, much longer time. This is also a really nice example of an answer describing the process of natural selection. So you could take this answer and apply it to whatever question you have. So, for example, maybe you have a question about tortoises and how some tortoises have short necks and some tortoises have long necks. And now you could apply the same answer to that. But instead of talking about cacti, you talk about the tortoises. Okay. Then another point on evolution is the idea of speciation through geographic isolation. So this is where a new species forms over time because the species become separated geographically. So either they end up on totally different islands and the two populations are separated from each other, and they're separated for so long that they become two totally different species. That's shown in the example of Darwin's finches, which are these birds shown here at the bottom. And there are a number of different species of these finches that are found on the Galapagos Islands. And so different islands have different food sources, and over time we've seen speciation, and these birds have changed so much that they are now separate species. So firstly, what is the definition of a species? Well, species contains individuals that are genetically similar to one another, and very important that they are able to interbreed and produce fertile offspring. So they can breed together, make offspring, and those offspring are able to have their own babies one day. So how does the process of speciation take place? Well, a population becomes separated, it gets split by a geographic barrier. So either a mountain comes up or a river starts running through the area and it divides this population into two. And in this particular example, let's go for an example. Let's do bunnies. So we've got rabbits in a field and there's a river running through the field. So what's going to happen now is these rabbits can't swim. So the rabbits on either side of the river have no contact with one another. They're not allowed to interbreed, and the two groups are not sharing any genes with one another. Now the environment is slightly different on the different sides of the river, and so these different populations are exposed to different environmental conditions. Natural selection is going to occur independently on each population. So what might be a favorable characteristic for survival on this side of the river might not be the same as on the other side of the river. So that's what we mean by natural selection occurs independently, so that the individuals in the two populations over time are going to become genotypically and phenotypically different from one another. And even if these populations were able to mix again, so let's say this river dries up and the two groups can come into contact again, they are no longer able to reproduce with one another because they have now become so genetically different that they are two different species. So we use the process of natural selection to explain how a new species is formed when one big group of animals or plants is separated by a geographical barrier, like the sea, a river, a mountain, or a lake. So again, this is a general explanation that can be applied to any specific question. So you can apply this to any particular animal or plant and the situation of how it was separated geographically by the sea, a river, a mountain, a lake, for example, and how that led to the formation of a new species. So keep that in mind. Again, like 6-mark question, so pretty good place for us to score some points. All right, let's just end off with a few general things. So we need to understand the scientific method, and you will be asked a scientific method question where they will give you generally the results of some kind of experiment. And so we need to be able to interpret what has occurred in an experiment. So we need to be able to identify the variables. We've got the independent variable. That's the independent starts with an I. So I changed that. That's the thing that the scientist who's carrying out the investigation is changing. Always appears on the x-axis of the graph. Then we've got the dependent variable, which appears on the vertical or the y-axis of the graph. And this is the outcome that we're measuring. So this is the thing that we're measuring. Then we need to make sure that all other variables are kept constant throughout the experiment. So let's think about an experiment. So let's say we are looking at the effects of light intensity, the amount of light, on the rate of photosynthesis, so how fast the plant is making food. So in that case, our independent variable, the thing that I'm going to change, is I'm going to look at different light intensities. The dependent variable is going to be the measure, or the outcome is what is the rate of photosynthesis. But then we need to make sure that all other variables are fixed or controlled. So we know that photosynthesis is not only controlled by the amount of light, it's also affected by the amount of carbon dioxide and the temperature. So we need to make sure that the temperature is kept constant and the amount of carbon dioxide is kept constant as well. So that the only thing that is changing and that can impact on the rate of photosynthesis is the light intensity. You need to be able to formulate a hypothesis. A hypothesis is a prediction of what you think is going to happen. Your hypothesis needs to include your two variables. So that's why it's important that you're able to identify the independent and dependent variables. Your hypothesis needs to then state the relationship between the two variables. So an example of a hypothesis for our photosynthesis example would be an increase in light intensity will increase the rate of photosynthesis. So there we've talked about a change, increase in the independent variable, which is light intensity. And then we said the outcome. What is the effect? We think it will increase the rate of photosynthesis, which is our dependent variable. And so you just guess what you think might happen. Your hypothesis does not have to be correct. It is just a statement that you are going to test, and you are going to see, does your evidence, does your experiment support your hypothesis, or does it not? Do you need to try a different hypothesis instead? Right, leading on from that, two important statements. We need to know how to make an experiment valid, and we also need to know how to make an experiment reliable. So validity is looking at, have we actually tested what we want to test? So if they ask you how you can increase validity or make an experiment more valid, if you see the word valid, what they are asking you for is examples of the fixed variables. So what are the fixed variables or controlled variables that you need to keep the same throughout this experiment? So if we're looking at our light intensity and photosynthesis example, how we keep validity, how we maintain validity and make sure what we're testing is actually what we're testing, is by keeping the carbon dioxide concentration the same and the temperature the same. Then finally, reliability. Reliability is, if someone else were to carry out the same experiment, could they get the same results or similar results? Obviously, it's not going to be exactly identical, but would they see a similar trend in their results? And so how you increase reliability, the two main ways are to repeat your investigation. So to do your experiment a second time and see, do you see the same sort of outcome again? And then the other thing that you can do is increase the sample size. So let's say you tested 2 plants for this photosynthesis experiment. Instead, look at doing 10 plants. So you've increased the sample size. And if you do the same experiment with 10 plants, do you see the same results as what you saw with just the 2 plants? So reliability, how you increase or make something more reliable, is the same for any experiment. However, validity is different because your fixed variables will be different depending on what the experiment is that you are carrying out. All right, so that was some revision points on some general stuff, as well as looking at evolution and genetics. So I wish you the very best of luck with your studies. Go out there and make some summary notes because there is so much content in life sciences. It's really important that you focus it down to the key points that you need to know, as well as practice lots of past papers. So past paper questions are available online, or ask your teacher. And practice, practice, practice. It's really good because you get an understanding of how the questions will be asked at the end of the year, as well as it gives you a really good idea of how they're looking for the answers. Please be really specific when you're answering the questions. The people who will be marking your exams are looking for very particular answers. So good idea to get used to that. All right, so I wish you the very best of luck with your studies, and I hope that your exams go extremely well, and good luck out there for your futures. Thank you so much, and goodbye.